Okay. Yeah. So yeah, I'll go straight into the project. I mean, I won't be talking about all the backgrounds on microgravity just because of last speaker, but also I know people are very hungry right now. I'll be fast. We're very interested in this particular long model that in this project. So basically, instead of the conventional model that are based on the upper airway modeling, we are interested in the distal lung here, or the alveolar lung, which is actually very complex in its structure and shape, having all the air, sacs come together in the way that the air can actually flow into in different compartments to allow this gas exchange. So in a conventional sense, I mean, there are many devices that are based on this conventional setup, which is having a membrane in the middle, which is being stretched in a way that it can brace. But then the issue with that is that, I mean, if you look at all the devices, I mean, it's still a flat surface, meaning that you can only basically model a relatively flat section of the lung, which is actually more useful for modeling the upper airway in the conventional sense, but not in the sense that if you want to actually recapitulate this very complex sac-like structure in the distal lung. So what we were actually working on, which is the basis of this project, is actually we back a few years ago we actually designed a system that's called a inversible scaffold device, which is basically having a pretty interesting structure by a templating process. Basically, what you do here is that you actually make these particles to begin with, and then put them together into lattice, and then you infiltrate the scaffolding material in between these spaces of the materials. Then you can solidify this scaffolding material, and then eventually selectively remove all the templating microparticles. So eventually create scaffold system that is very structurally mimicking or similar to the actual distal lung, which has all these sacs come together or pores come together with windows connecting different pores like gas exchange. And then what happens here is that if you then seed these epithelial cells or distal lung epithelial cells into the scaffold system, you can see how the epithelial cells are able to populate the entire surface of these structures, forming a very nicely biologically relevant distal lung model in the pattern. Let me try to go back and then see. Okay, so this move it in place. So basically, if you look at this confocal image, not only cells are lining very nicely along the surface, but also you can see the windows that are actually connecting this, the adjacent pores, serving as the windows to allow gas exchange, all the other functions happening in this lung. We did show that these cells were actually nicely expressing the biomarkers, for example, ZO1 in between the cells, the junction proteins, but also if you do the cross-sectional view, this is actually standing on the bottom right. Basically, you would see that the epithelial cells were able to actually form very nice single layer lining along the wall, but also the windows connecting again the different pores. We did show that if you culture these cells in the scaffolds, I mean, they would be able to stay alive for, in this case, we'll show up to 2 weeks. But then in actual experiments, we show that you can actually culture this for up to months of time without many dead cells in the system. And what's interesting here, I want to emphasize that because we actually make the scaffold system with hydrogels, in this case JAMA, we can actually make the mechanical property very nicely matching the actual mechanical property of the actual distal lung, which is in the range of a few kilopascals, which is actually very different than the conventional devices that are based on, for example, PDMS, which is actually much more stiffer than the actual distal lung. So it's a very complex slide. I won't go into everything here, but I want to actually emphasize 2 points. So one is that if you compare our model, 3-dimensional JAMA-based system, with the 2-dimensional gel, same material, same stiffness, actually see some differences. In our model 3D JAMA, the geometry actually matters because the cells were able to actually populate the entire surface of the scaffolding system without leaving any empty spaces. But then if you see the same cells on the 2-dimensional JAMA, you would actually only see patches, and somehow the cells wouldn't be able to connect themselves together into a confluent layer on 2-dimensional surface. But then what's interesting here is that it's also different if you compare 2-dimensional JAMA, this one, I mean, the stiffness of the actual the distal lung, but then with 2-dimensional PDMS, which is much stiffer, you also see differences indicating that the geometry, but also the stiffness of the matrix actually matter in coordination to really allow you to produce the proper lung model in this case. And then you can combine that with the conventional, designed system, the chip device to allow not only delivering the oxygen, the nutrients from the bottom channels, for example, but also the side chambers to allow mechanical actuation or breathing events of the lung in this case. In the bottom left image or video, we can see that this is actually lung, distal lung that's being breathed at the normal pace, which is about every one breath every 2 to 3 seconds, and also at about 10% oxygen level, which is physiologically relevant. We showed that, the lung model would actually keep their TO on the breathing, which is totally expected. But what's more interesting here is that with breathing or mechanical actuation, you would actually see an enhanced expression of the CO1 biomarkers, which is indicative that mechanical breathing event actually increases the physiological relevance of the model. So based on that, this is a very interesting model, but then the issue with that is that we didn't have any vaccination there, right? So, or immune cells in there. So based on that, we actually designed this tissue chips in space project by expanding that into the model that's more physiologically relevant. So we're working with Dr. Vinsha Gu from UCLA, who is going to actually provide us with all the organoid-derived cells to allow these different cell populations to be built into the model, but also have Space Tango as the implementation partner to allow us to miniaturize everything into a space-ready format. So we have 3 aims. Aim 1, basically, again, is focused on vaccination of these models, basically building vasculature that's perfusable within the model that allows creation of the epithelial and endothelial junction to allow the oxygenation, but also the other functionality to be built into the actual system. And aim 2 is that once you have the vascularized system, you can start to also build immune components into these models. For example, in this case, we're thinking about having both resident immune cells, but also more importantly, the circulating immune cells that can be actually perfused within the vascularized lung-chip model to allow recapitulating of both healthy and also potentially fibrotic phenotypes of these models of these lung tissues in the model. And then we're going to be, of course, hopefully be flying these models into the space station in aim 3. I will probably not go into details here because, Dr. Joseph Wu also mentioned Space Tango in the first talk. So, they have been working on a number of different missions, very experienced, including some of the previous chips in space program. But then, again, we're working with them. We're trying to really hopeful to bring our previous model in a larger form factor into a small form factor that can be actually fit into this cube lab, which is going to hold up to about 6 chips per mission for the actual experiments. We're going to be automating every operation, and we do have capacity to hopefully also incorporate a few sensing modules. We do now have a glucose sensing module right now ready with Space Tango, but also we're hopefully to do this project, jointly develop some of the oxygen sensing modules for the purpose as well. And then again, we're looking at about a couple of years from now, hopefully getting our first flight in the year 3 of the project, but then hopefully taking from there, we'll have another chance to fly again in year 5 of the project in the next phase. Before I end, I want to give a few slides to really talk about some of the recent developments of the project. So basically, what I mentioned is that, we have been now using some of the primary cells that are commercially available for making these models. And working with Dr. Mingxia Gu, we have been trying to actually use organoid-derived cells, just because, for these organoids, you can actually have all the different populations of cells that are being generated within the organoids, including the epithelial cells and, of course, all the endothelial cells that are of interest for this project. So what happened here is that in the conventional sense, right, if you really create these organoids, in this case, for example, the vascularized organoids, I mean, they are really nice in terms of structure, but then you cannot really structure these different populations of cells in the right architecture because, I mean, they're all different across different fabrication batches of the organoids. So in the previous example, what we did together was that we actually isolated these different single cells into the populations of cells into single cells, and then we reseeded these different cells into the scaffolds that I just mentioned before. So what happened there is that it's very interesting that we did say that these cells would be able to reorganize themselves according to the structure of the scaffolds by having the epithelial cells first lining on the surface of this scaffolding system, but then followed by the epithelial cells then that are sitting on top of these endothelial cells, forming very nice structure that's quite similar to a mouse lung structure at embryonic day 2.5 in vivo, so indicating that you can potentially use the scaffold system as the guiding point to allow reorientation of the cells in the way that you can reproduce very similar structure. So that's one update, but then again, this is basically reseating the scaffolds all together on top of the scaffold system, but then we have also been working very diligently on further creating a chip device that allows you to really vascularize the lung model at a larger scale. So this is canonical multi-chamber chip device that has all these different posts to allow sterilization. And in the first example, for the work we have been doing the past couple of months, we have shown that you can potentially endothelialize the entire chamber quite nicely. And then in the preliminary experiments, we did show that these endothelialized vessels are actually potentially perfused. We can actually perfuse this microparticles from one side, and then you can see actually some of the particles actually going through the entire structure of the vascularization. So again, that's the first step, and then we're now trying to integrate that with all the organelle cells and also the epithelial cells in the sac-like structure, as I mentioned in the beginning. So I'm going to stop there, and then thank you again for this opportunity, and very excited to be looking forward to our potential flights in the next couple of years, and thank you very much.